Hello and welcome to August Longevity Review, where we will take a look at the first human rapamycin trial devoted to longevity, a novel autophagy activator which might be even better than rapamycin, how human umbilical cord stem cells improve various biomarkers in rats, a review of life-extending combination treatments in mice, a look at the 2024 Aging Research and Drug Discovery meeting, and finally, in the Canadian content section, a new DNA repair mechanism found in the world's hardiest organism. This review is presented to you by the Canadian Longevity Association, Canada's premier longevity nonprofit. My name is Chris Linnell, and I'm the founder and president of the association. If you'd like to find out more about what we do, then please check out the links below this video. And also, don't forget to like this video and subscribe to our channel. We've got a lot to get through, so let's get started. I've waited 3 years to bring you the results of the first study we're going to take a look at, since 2021 to be precise, because I helped with the crowdfunding campaign whilst volunteering for lifespan.io. I helped write the crowdfunding page for the Pearl trial, the participatory evaluation of aging with rapamycin for longevity. It raised over $180,000 from 409 backers, including myself, and was the very first rapamycin trial in the context of longevity. It was organized by Dr. Sajjad Salzala and Ageless RX. All I had gotten up until now was my reward t-shirt, so it was great to finally get the first results for this trial. So let's take a look at the first result. Now, the first thing I should mention is that this is still a preprint. Now, what does that mean? Well, as it says here, this article is a preprint and has not been peer-reviewed. It reports new medical research that is yet to be evaluated and so should not be used to guide clinical practice. Basically, the researchers have written the first, or in this case, the second draft of their paper. However, other researchers have not yet read it through it and checked it for mistakes or inaccuracies. This paper could still be revised before it is finally published. With that in mind, let's take a look at the study design. So around 1,000 people were assessed for eligibility, of which 125 were chosen to participate in the trial, and they were split into 3 groups, 40 of whom received 5 milligrams of rapamycin once per week, 35 received 10 milligrams once per week, and 39 acted as the control arm. The trial lasted for 48 weeks. Also bear in mind that this was taking place during COVID. The primary endpoint of the study was changes in visceral fat as measured by dual energy X-ray or DEXA scans. Secondary endpoints included changes in lean tissue mass and bone density as determined by DEXA scan, as well as changes in blood biomarkers from complete blood count, blood electrolytes, liver function, renal function, serum glucose, insulin, hemoglobin A1C. Standardized self-reported surveys of quality of life and frailty were also completed by study participants. Before looking at the results, we should talk about the first rather unfortunate finding, which was that the dose given wasn't actually the intended dose. After the trial had already started, the researchers discovered that the compounded form of rapamycin used was only 28% as bioavailable as the generic or sort of normal form of rapamycin. Now, the reason they used a compounded form is that rapamycin has a distinctive shape. It's sort of triangular, a rounded triangle, and they didn't have the money to be able to manufacture a placebo that looked the same. So to keep the form of rapamycin the same between the placebo and the 2 treated groups, they used a compounded form. Now, unfortunately, once the trial started, they found this out, which means that instead of 5 milligrams and 10 milligrams, the participants received either 1.4 milligrams or 2.9 milligrams respectively, so considerably less than the intended dose. So it was a very low dose rapamycin trial. Now, this finding and further investigation is the subject of the first paper down here. Now, I'm not going to go into that today. I've pretty much just sort of explained it to you right now, but you can read it. And again, there's a link below this video. But we will continue by looking at the results of the trial itself. First, we'll take a look at the side effects or the adverse effect summary, which is down here. starting with the bar graph at the top here, with the serious side effects being the blue bar, minor ones, the orange bar, and withdrawals from the trial being the black bar, with 10 milligrams, 5 milligrams in the placebo group in that order. You can see, first of all, looking at the withdrawals, that there were a lot more withdrawals from the placebo group, there being 6 in total, compared to the 10 milligram with 3, or the 5 milligram group with 2 withdrawals. So sort of a slight win for the trial here. And similarly, it goes with the more serious adverse effects, there being 3 in the placebo group, 2 in the 5 milligram group, and only 1 in the 10 milligram group. So again, a sort of slight positive result for the trial. And looking at the more minor ones, we'll go the orange bars are the same as B here on the bottom left. So we'll take a closer look at that. And there were a total of 117 minor side effects with the 10 milligram group, 116 with the 5 milligram group, and 122 in the placebo group. So once again, the trial did maybe even slightly better than the placebo group in total. And the bars were divided between male and female, females being blue here. And since there were a lot fewer females in this trial than men, roughly 30% of the trial participants were female, the percentages actually, in terms of these adverse events, were pretty much the same between men and women. There was no real difference. So these two bar graphs are showing on the left is the total number of events in total, and on the right is the number of participants who experienced these events. And again, there was no real difference between placebo and 5 milligrams or 10 milligrams. So again, demonstrating that the safety profile of rapamycin was pretty similar to that of placebo, which is obviously a positive thing. And most of these adverse events, as I say, they were minor, mainly sort of gastrointestinal issues or canker sores, which is a known side effect of rapamycin. But again, in such instances, the dosage can simply be reduced and then slowly raised up again to get rid of most of these minor events. And also you have to consider the fact that this is over the course of 48 weeks, almost a year. And over the course of a year, people are going to experience sort of minor ailments, colds, and such like. So if people are sort of watching out and noting down what's happening with themselves, then there will, of course, be side effects. People will experience things over the course of a year. So this is not a cause for concern in any way. It's the same pretty much with placebo. Next, we'll take a look at 4 of the main endpoints, which were changes in visceral fat, lean tissue mass, and bone mineral density and bone mineral content. They looked at changes in these measures after 24 weeks and 48 weeks, with the black bars being the 10 milligram group, the orange bar being the placebo, and the sort of yellow orangey bar being the 5 milligram groups. And you see A and B here. On the right-hand side, it's showing all the individual participants denoted by these dots. And on the left is the percentage change. And starting with visceral adipose tissue, so that basically means the fat around our organs, like the belly fat, the more dangerous variety of fat. And although it seems that they all increased, the 10 milligram placebo and 5 milligram groups, they did not actually reach statistical significance. The same went with bone mineral content, changes in bone mineral density, and changes in lean tissue mass. So we won't spend time on this because the authors then divided the participants into men and women to sort of get a more granular view of what was going on. And there they actually found some significant difference in at least one of the measures. So this first chart here, once again, is showing all the individual participants, but we'll look at the percentage change, which is here in figure 1. So they flip the order around here. So we're starting with changes in lean tissue mass. And what these bars represent, black bars are the 10 milligram females after either 24 or 48 weeks. Turquoise is the placebos. Light blue is the 5 milligram group. Again, this is all females. Then for the men, the brown is 10 milligrams. The yellow-orange is a placebo, and the pink is, or the light pink is the 5 milligram group. And Here they saw a significant difference in lean tissue mass for the women doing 10 milligrams. So this reached statistical significance. You can see here these bars are quite a bit bigger than the other bars. So there's no real difference in any of the other groups. And lean tissue mass is something that we want, that sort of muscles. More lean tissue mass is better. So this was a positive result for the rapamycin trial. As you can see, there was at least a 5% difference in or increase in lean tissue mass after 48 weeks in the 10 milligram female group. In terms of visceral adipose tissue, so again, the fat around the organs, although it looks like the bars changed considerably, this did not reach statistical significance. Perhaps the 5 milligram males saw a slight reduction, but that again, after 48 weeks, that really didn't change much from baseline. 5 milligram females and placebo females saw a much bigger sort of increase in visceral fat, which is obviously not good. Same goes with 5 milligram males. And although the 10 milligram female saw a reduction after 24 weeks, it sort of rebounded after the 48-week mark. Now, looking at changes in bone mineral content and bone mineral density, since they're showing pretty sort of similar things, higher is better. We don't, we want to have more mineral content and density in our bones, which means our bones are stronger. And it did appear that for the women, both for bone mineral content and bone mineral density, there was a drop in that, which might be slightly concerning. But again, this did not reach statistical significance. Same goes for the men up on the 5 milligram side, but the men on 10 milligrams saw an increase in both bone mineral content and bone mineral density. But again, I have to emphasize that this did not reach statistical significance. Before looking at our final set of graphs, we'll just briefly talk about the blood work. Now, for most measures, there was no significant change. However, there was some change on 3 of them, namely hemoglobin A1C, which measures your blood glucose over the preceding 3 months, calcium, and CO2 content. And they found that while all changes we observed remained within healthy range, hemoglobin A1C saw a small increase at 48 weeks for the 5 milligram cohort of men only, but the glucose and insulin levels, there were no significant changes in them. For calcium, there was a significant change in the 10 milligram and placebo groups for men, saw a reduction in calcium. And for CO2, they mentioned that interestingly, carbon dioxide levels decreased only in the treatment groups, which was mainly seen in the 10 milligram group, which steadily decreased carbon dioxide levels over the course of the study. And for that, there was no difference in gender. Next, for a small subset of the participants, 24 in total, 9 women, 15 men, they also gave them an epigenetic age test by True Diagnostic. And there they found no meaningful significant changes between groups. They also did a gut microbiome analysis via the gut health test from Thorne from 81 of the participants, 31 women and 50 males, where once again, no significant differerences were observed. However, drilling down a little bit further, they noticed small significant increases after 48 weeks in gut dysbiosis in males in the 10 milligram treatment group. So I guess disturbances in the composition of the gut microbiome, and trends of increased intestinal permeability in females in the 10 milligram groups. Both of those things we don't want, but they're not sure how significant these findings are since it was only a subset of the trial. So again, it makes it more difficult to get any sort of statistical significance or meaning out of it. Therefore, larger sample sizes in future studies would lend more meaningful insight into these measures. Finally, they looked at perceptions of health from the participants. First up was the WOMAC pain score. Now it says SF36 here, but I believe that's a typo. Again, a reminder that this is a preprint, so there are probably going to be some mistakes in it, which would be corrected by the time it actually gets published. And once again, this is divided by gender, with the 10 milligram female group being in black, turquoise being placebo female, light blue being the 5 milligram female, brown being the 10 milligram male, yellowy orange the placebo male, and the sort of light pink being the 5 milligram male. And higher is better. And there was a significant change in the 10 milligram female group. You can see here on the left with these two stars denoting the statistical significance of that. So the perceptions of pain in those women improved. There was a slight improvement also in the 5 milligram women, and no real change among the men, certainly after 48 weeks. Again, so none of these scores were significant, but again, 10 milligram group seemed to have a lot less pain, such as arthritis or things along those lines. Now, the SF-36 is sort of a survey on general well-being, and that can also be divided into emotional well-being and just general health score changes, self-perceptions on that. And this one wasn't divided by gender, since there was no real difference between the genders in this. And here, emotional well-being score changed significantly after 48 weeks for the 5 milligram group and the placebo group. No major change for the 10 milligram groups here. For the general health score, so it was in the 5 milligram group that there was a significant change compared to baseline for the 5 milligram group. For the 10 milligram placebo, there was no specific or no significant change. And there we have it. So I must admit to being slightly disappointed that there weren't more significant positive results, the only ones being increases in lean tissue mass and lower perceptions of pain amongst the 10 milligram female group. But I guess that is mainly down to the fact that the doses used were considerably lower than intended. And that especially goes for males, since in mice at least, rapamycin has a much greater effect on female mice for any given dose compared to male mice. However, the important takeaway is also that the safety profile was comparable to that of placebo at this low dose. Obviously, we need to see further trials with higher doses to get a much better appreciation of the effects of rapamycin in humans. I'd be very interested in hearing your thoughts about this trial. There's a link to this trial below this video, and please share your thoughts in the comments below. Nonetheless, this was an important trial and marked a good first step in our understanding of rapamycin for longevity in humans. Moving from rapamycin to a new molecule which might be even better than rapamycin. So CXM102, as it's snappily called, was developed to increase autophagy, which is the sort of the recycling mechanism within cells to try to rejuvenate the stem cells inside bones, to reduce bone loss and to perhaps prevent osteoporosis, because I think previous studies have found that increasing autophagy kind of rejuvenate those stem cells, and has some other interesting effects as well, which we will shortly get into. We'll start by looking at the first figure, or graph, I should say. And here on the left, you can see the chemical structure of CXM102. And the really cool thing about this study is that they used rapamycin as a positive control, because rapamycin as well also increases autophagy. It's one of its main mechanisms of action. And firstly, this is now in sort of cells and cell culture, so not in animals, but later they actually do move into mice, which is where it gets really interesting. But firstly, they wanted to see whether or not it increased autophagy. So on these pictures here, simply put, the green indicates more autophagy, basically. And you've got on the left the controls, middle rapamycin positive controls, and the CXM102 new molecule on the right-hand side. And on the top, you have Old animals. In the middle, you have old animals plus chloroquine, which reduces autophagy. At the bottom, you have young animals. And I should say the old animals were sort of 14 to 18 months old. Oh, these aren't animals, these are cells, but I guess they came from old animals. The old animals were 14 to 18 months old, so between the equivalent of late 40s to late 50s in human years. Basically, green indicates more autophagy. So for the control, the old cells, little autophagy. Chloroquine, again, little autophagy. In the young cells, lots of autophagy. Rapamycin increases it in the old animals. However, CXM102 increases it even more. You can see by there being more green there, which is also shown by this bar chart here on the left. This is showing levels of autophagy and higher bars showing more of it. Control being old, RAPA here, CXM 102 and young cells. And once again, CXM was performed better than rapamycin, not as good as the young, but certainly a lot better than the old controls. And likewise on the right-hand side, similar type of results. Again, green being CXM 102, blue being RAPA, black being the controls. Higher is for the most part better. And for most of these, CXM 102 was not only better than the control, but better than rapamycin as well. Now, that was just in cells, and the next one will also just be in cells. They're looking at its effects on senescence. So senescence is a bad thing. Here, SA beta gal is a marker of senescence, and it's kind of hard to sort of see here, but the greater concentration of CXM 102, the less senescence there was. So senescence is shown by, I guess, a darker blue color in these pictures here, and higher doses led to less senescence, which also can be seen here in this bar graph. Once again, markers of this SA beta gal, and the higher dose, the less senescence there is compared to control. And yes, on the bottom, also here, again, this is in cell culture, not in animals. Interesting, on the left here is osteogenesis, so that's bone growth marker, so more red is better, and compared to the control, CXM 102 produced more bone growth. Phytogenesis is fat growth, and which we don't want in bones, because in bones, especially the bone marrow, you've got both fat and bone formation, and as you get older you get more fat and less bone, which is not a good thing. So here red again indicates the fat cells, and under control there's more red. With CXM 102 there's less red. That's a good thing. Next, just briefly, they looked at something called TFEB, which helps causes the generation of lysosomes. So lysosomes are the sort of part of the cell, the organelle in the cell, which acts as a recycling unit, sort of the furnace. It destroys proteins, so they breaks them down so that they can be then sort of reused to produce better, newer proteins. And TFEB produces more lysosomes, and here I won't go into it, but as the dose of CXM 102 increases, the levels of this TFEB increases as well, indicating that more lysosomes are being formed. Next, once again, in Petri dishes, just briefly take a look at this. This is once again showing the difference between fat and bone formation. And here, I shall go into it briefly. You can see here, top line, VP stands for verteporfin, which supposedly stops bone growth, gets used to prevent malbone formation, or bones growing where you don't want them to. And CXM, obviously, they hope to increases bone growth. And once again, the staining red indicates more osteogenesis, so more bone growth, and minus sort of normal bones, so don't have either of the substances. There's some bone growth. CXM really makes a lot more bone growth here when it's only it. If there's no CXM and this VP stuff, there's even less bone growth than the control. And when the two are combined, CXM 102 rescues it to some degree, not as much as on its own, but still better than before. Now it gets interesting, because we're moving away from Petri dishes or test tubes into live mice, and they used, again, mice from the age of 14 months, which is roughly someone in their late 40s, and they were given this drug for 4 months, and they were injected with it, and so at the end of 4 months, after 18 months, they're roughly in their late 50s age-wise, though of course mice and humans aren't quite comparable in that way. So you can see the time across the bottom here in terms of months, or just months from start of treatment. And at first we're looking at bone mineral density, and red is just the old controls, green is OC, which is the CXM102, OV is this verteporfin, which stops bone growth, and OVC is a combination of both. And certainly on its own, it has much higher levels of bone mineral density, though it should be said that it started at a higher level of the mice given the treatment anyway. Here's a percentage, I guess, change in bone mineral density, and it performed much better than the old controls. And when combined, and OVC performed better than OV as well. And similarly here, another sort of markers of bone density, the green bar being the treated animals, and it's much closer to the young controls compared to the old controls, with black and red respectively there. Then we have some visual cross sections of the bones, and you can sort of see here young bones full of whole bones, or this big gap in the middle lacking a bone. With OXM 102, there's a lot more bone in the middle. They're not as hollow. The OV, it's again even less bone, and adding the CXM 102 rescues it a little bit. And here is again sort of the cross sections of the bone at different points, and just showing that there is more bone in the treated animals. Again, black being young, red is old, and green being the treated animals. Bars higher than the control, not quite as high as the young controls. And this is sort of just three different places in the bone where this is tested in all three places. The treated animals have a thicker bone, basically. And here is showing, I guess, sort of a separation. Couldn't quite get what this meant exactly, but I guess less separation is better. And once again, the treated animals had less separation than the old, but not quite as good as the young. But then in coming to how strong the bones are, so this is sort of fracture on the F here, how much force is needed to cause the bones to fracture. I guess this is after the animals were euthanized and their bones were taken out and tested, I guess. And once again, we can see here that the treated animals did a lot better than the old, not quite as good as the young for fracture risk. This is stress response, how much, I guess, stress they can handle. And on the right here is how stretchy the bones are. Young's modulus. I've never heard of that before, but that's about how much something can stretch, I guess. And once again, the treated animals do a lot better than the control. Not quite as good as the young. Next, they looked at, once again, the composition of the bone in terms of bone and fat. Won't go into this in detail, but you can see here on the sort of the left, you got young, old, and the treated animals. And the white is showing, I guess, fat deposition, and the treated animals have less fat than the old controls, though more fat, I guess, than the young animals do. But now we get to the really, sort of skipping over that. Now we get into the really interesting one, the figure that made me choose this study to be included as a highlight, because we have a lifespan study, and we once again are having rapamycin as the positive control. Now, before I go into this, I should say that there were very, very, very few animals used in this. In fact, for both control, the treated animals, and the rapamycin treated animals, there are only like 5 or 6 mice in each of these, which means that what I'm about to show you could be a statistical anomaly. And on its own, I would not really trust at all. So you can see here from 14 months, the mice were injected either with rapamycin or CXM 102 or DMSO. That's just sort of a kind of saline solution, I think, as control. So they all the animals were injected so that they all suffered the same, so to speak. And from 14 months of age, then they were led to live out their lives. And the red line is the control, and they died, here. Blue line is rapamycin, and the green line is the CXM 102. And they said that rapamycin, the average lifespan extension of rapamycin was only 5.6% more than control, whereas the CXM 102 mice live 22% more than control. So, but again, such few mice that this really could be statistical noise. You really need a lot more mice to really get a proper lifespan study. But what I'm about to show you gives a little bit more credence. I mean, the authors do say that they need to conduct larger studies, and I believes they will be doing so to get see if this sort of holds true with more animals. But the next things we're going to look at give a little bit more credence. So first of all, B here shows serum levels of tumor necrosis factor alpha, a marker of sort of inflammation. So 14-month-old mice on the left, the red showing old mice, that's after 6 months. So they're 20-month-old mice, and levels of this inflammatory marker have gone up considerably. However, OC, the treated animals, it's considerably less, statistically significantly so. And rapamycin reduces it, but not to the same extent. Here's another one, IL-1, another inflammatory marker, and it rises in the mice. The old control mice, it doesn't rise as much in the treated animals. And again, it performs better than rapamycin on the right-hand side here. Likewise with IL-6, same thing again. Older mice see rise in this inflammatory marker. The rise isn't as much. In the CXM 102, again, it performs better than rapamycin here. Now, these images, the top line refers to the liver, and the staining with oiled red, this is used to stain fat cells. So you can see from 14 months to 20 months, the number of fat cells and the amount of fat increases quite a lot. Less so with the treated animals or rapamycin, but the treated animals are even better than rapamycin. There's less red here than in the rapamycin, but both are better than the control animals. Now we move to kidneys, but this time it's looking at fibrosis. So again, with age, fibrosis increases, and I guess the blue color means more fibrosis. So as the animals age, they get more fibrosis, and both treatments, CXM 102 and rapamycin, don't produce less fibrosis. And now we look at the heart on the bottom line here, which again, even more dangerous. If the heart gets fibrosis, it can't function as efficiently. I mean, the same goes with the kidneys actually, but still here it's more visible in the heart. You can really see as the animals age over 6 months, they get a lot more fibrosis, and this is drastically reduced than the CXM 102 and rapamycin treated animals, though we'll see in the bar graphs shortly that the CXM performs better than rapamycin, which is what we see here. So on the left-hand side is number of lipid droplets, so fat in the liver. You can see from 14 months to 20 months, it rises massively. Hardly any rise in the CXM 102 animals. Rapamycin rises considerably more. Now, the number of 5G and H are both from the kidneys, and here's the fibrosis marker, and it rises a lot between 14 months and 20 months, rises, but not as much in the CXM 102, and which performs better than rapamycin. And H here is sort of diameter of the renal sort of kidney tubules, and bigger is better. And you can see here that it decreases as the animals age in the controls. That is prevented to a large extent with the CXM 102 animals, whereas rapamycin doesn't really do much better than the old controls. And finally, here looking at the heart, again, from these images, we can see in the bottom here, fibrous fibers or fibrosis increases a lot over 6 months, hardly increases in the CMX 102s, and which again performs better than rapamycin. So it's the fact that at least in these indicators that CMX 102 performs better than rapamycin, that leads a little bit more credence to this lifespan study. But as I said, and as the authors themselves acknowledge, they need much bigger studies of this new molecule, but interesting nevertheless. So something to keep an eye on, especially if it has such a snazzy name like CMX 102. Moving from a molecule which rejuvenates stem cells in mice to injecting actual stem cells into rats, the next study used human umbilical cord stem cells or mesenchymal stromal cells, basically the same thing as stem cells. So very young cells, obviously coming from an umbilical cord. And the authors claim that this was the most thorough examination of the effects on old rats of these very, very young human stem cells. Now, going to go down of here we are looking at the study design. They with the treated rats, they injected them once a week for 4 weeks before then euthanizing them to take samples. There was also old control rats and also young control rats, both of which were just injected with saline. And the old rats were 24 months old, which is human equivalent of 60 years, though of course a human is not the same as a rat. And these graphs here just to show that it was the correct type of cell that was injected into the rats. And these stainings here just show that the injected stem cells led to bone growth, osteogenesis, fat growth, petogenesis, and chondrogenesis, so chondroitin, what is used in human or in joints, basically. Now, next, gonna need to cycle down a bit further. Here we go. So first of all, after the rats had been euthanized and then sampled, they took a look at the senescence profile of the rats, or the senescent markers, I should say. And it's a bit hard to see on these cross sections. So we have lung, kidney, and then on the bottom left, intestine. It's a bit hard to see from these cross sections exactly what is going on. I mean, these are markers for senescence. P16, P21 are senescent markers. However, in the intestines, you can just looking at this a cross section of the intestine, and you can see the age controls are in the middle, young on the left, treated animals on the right, and just visually, the treated animals, their intestines, the villi in the intestine, look closer to that of the young animals than the old control animals. Now, here are bar graphs showing these two senescent markers, P16 and P21. And in all of these, the black bars are the young animals, red bars are the old controls, blue bars are the old treated animals. And for all in lungs, kidneys, and intestine, for both P16 and P21, the treated animals had much lower scores of these senescent markers, and it reached statistical significance. And just visually, you can see there's a big difference between the old controls and the treated animals. Next, they looked at other biomarkers in various organs. So beta-gal is another senescent marker, and once again, the lung, kidney, intestine, and peripheral blood, the levels were much lower in the treated animals. And again, for all of these and all of the other figures we're going to look at, black is young control, red is the age control, and blue are the treated animals. And just looking at it, you can see the blue bars are always lower than the red bars. And here is lipofuscin, which accumulates in tissue. We don't want it to. And once again, levels are lower in the treated animals compared to control. SOD, superoxide dismutase, an antioxidant enzyme. I think in our bodies, we want more of that. And in this case, the blue bars are higher than the red bars in the various tissue. Actually, in the lung, it looks like they're even higher than the young animals, but there you go. I'm not quite sure what that means. MDA is a mark of oxidization. So again, lower is better. And once again, the blue bars are lower than the red bars in all four tissues. GSH is glutathione. Again, a antioxidant sort of enzyme. We want more of that, but higher levels are better usually. And once again, blue bars are higher than the red bars. And in some cases, in the lung and peripheral blood, actually higher than in the young controls. IgG is immunoglobulin, immune marker of the immune system. Higher is usually better unless you have an autoimmune disease, in which case you don't want it to be higher, but normally higher is better. And here, once again, blue bars are higher than the red bars, so they don't, sometimes in the lung, they approach the same as the young. IL-1 beta and IL-6 are, again, inflammatory markers. We want lower is better. And yet again, blue bars are lower than red bars, though the intestine here only by a little bit, for example. Same goes with the kidney and the peripheral blood. I'm not quite sure why they've changed the way they're showing it. But anyway, in all cases, the treated animals had better markers for these biomarkers. Next, I looked at the brain. Top slide, again, showing senescence, darker, better. It's hard to really see here, but these images are showing Nissl in the hippocampus, which turns out they're part of neurons, and they're like an organelle inside of neurons, as I found out researching this. And the numbers seem to increase, as you can see by E here. Again, blue bar higher than the red bar, approaching that of the young controls. And according to the authors, their organization they weren't as disorganized as the aged controls in the treated animals, closer to that of the young. Again, here's senescent markers, P16, P21. But let's look at the graphs because that's a bit clearer than those images. And again, same as the other tissue we've looked at, P16, P21, senescent markers, blue, a lot lower than the red bars. Good thing. Beta gal, once again, blue is lower than red. The diffuskin, blue is lower than red. SOD, blue is higher than red. We want that. MDA is lower. Oxidization, again, we want that. GSH, again, higher is better. Once again, it's higher than the young, actually. Hopefully that isn't a problem. Immune response or immunoglobulin, again, higher than red. We want that. IL-1 beta and IL-6 lower than the red. Not by much, not statistically significant. There are none of these stars above. So yeah, and all these graphs we're looking at, the more stars they have, the more statistically significant they are, which means that there's less risk of the results just being due to chance. And moving to the next organ. This time it is the spleen, so part of the immune system, filters blood or replaces blood, and yeah, so useful for in terms of our immune system. And once again, these images aren't entirely clear, but the bar charts tell the same story as the previous organs. Once again, when we want the blue to be lower, it is. When we want it to be higher than the red, that is, it is. So number of apoptotic, so cells which are sort of dying, lower in the treated than the control animals. The rest, once again, the same as we saw before, P16, P21, all the same as we saw before. And in each of these instances, again, the blue bar is either higher or lower than the red bar, as we would want it, basically. And here's a distribution of two types of immune cells, CD4, CD8, and the ratio is important. We want a higher ratio of one to the other. Again, the blue treated have better ratios than the old untreated, though again they're higher than the actual young animals. So I hope that isn't a problem. The authors don't indicate that it is, but there we go. Next, which organ are we going to now? Okay, we have the liver this time, and the same story seems to unfold. Now, one difference here is one issue with growing older is getting fat deposits in the liver, fatty liver disease, for example. And oil red O is a staining method which shows fat, and more red is worse. And the age controls have more of this red staining than the young controls, whereas the age-treated animals have less of it, which can be seen in this bar graph on the right. The old controls have a huge amount, or a lot more than both the young and the treated animals. And once again, same senescent markers, P16, P21. Easier to see on the bar graphs than in the liver slices. And once again, we have all the same biomarkers we looked at before. And once again, the comparison between the blue and the red bars is always in a positive direction as far as we are concerned, or we would like. And finally, we get to the heart of the matter, or at least the heart of the heart. Here is the heart tissue examined. And once again, we start off with the oil red O staining, and you can sort of see that the age controls are a little bit redder than both the young control and the age-treated animals. Mason here refers to fibrosis, and the old controls have more fibrosis than the treated old animals, though not as though the young have quite a bit less. And again, with senescent markers, P16, P21, looking at the graphs, you can see once again the treated older animals have much lower levels than the old control. And you can see here the fatty liver, the fat staining, huge difference between the treated and control animals, though a lot higher than that of the young. And here's the cardiovascular fibrosis percentage, a lot higher in the untreated animals, and the area of that fibrosis, once again, is a lot higher than in either the young animals or the old treated, which looks a bit lower, actually a smidgen lower than in the young there, but that's not statistically significant. There we have it. One more organ, so to speak, at least the microbiome, not exactly an organ, but certainly a vital part of us. And this is kind of hard to really explain most of these, difficult to sort of interpret, except that the authors note a couple things. Just looking here on the top left, the blue dots, unfortunately, they've changed the naming of these groups. Group 1 is the young controls. Group 2 is the old controls. Group 3 are the treated animals. And one thing is the microbiome of the old treated and old controls are more similar than that of the young controls. However, for certain species of microorganisms, you can see here in D, in this middle bit, once again, on the left are the young controls, middle are the old controls, right are the treated animals. Although the treated animals are much closer to the controls than to the young animals in all instances, they are still better, closer to the young animals than the old controls are. So here, where the young animals have a lot of these microorganisms, the treated animals have slightly more than the controls. And here, where the young animals have less of these microorganisms, the treated animals have less than the controls. So again, all pointing in the right direction. However, the one thing to mention is that very few animals were actually used in the study. Once again, the authors say it was hard to find old rats, and they're going to try to redo the study with more animals as they sort of find them, so to speak. Also, they don't know exactly what caused this. They suspect it was exosomes from the stem cells which caused all these beneficial effects. But again, interesting that they used human umbilical cord stem cells. So if they worked in rats, you think they would work even better in humans. So as the author said, and I mentioned at the beginning, this was supposedly the most comprehensive evaluation of these umbilical cord stem cells in rats, and they certainly had a positive effect. Now we're going to take a look at a review paper. Now, normally I don't cover review papers in the highlights section, but when the review paper is about the best combination treatments used to extend mouse lifespan, I certainly make an exception. Now, there's a lifespan.io article about this paper, but let's dive into the paper itself. Now, I won't go into too much detail in this paper, as it is well worth a read for yourselves, and it's very long. Once again, the link can be found below this video, but I will briefly take you through it, and starting with the methodology used. Now, paper was written through the prism of the hallmarks of aging. So there are originally 9 hallmarks. A few extra have been added as of the writing of this paper in 2023. There were 12 hallmarks, and there are several other hallmarks proposed, which they sort of also included here. Now, the idea being each of the hallmarks, in case you're not aware, represent a different form of aging. It's a way of sort of look at aging, be it senescent cells, telomere attrition. There are, as I say, at least 12 of them, and they wanted to see which of those hallmarks each of these interventions primarily targeted, with the idea being to sort of treat aging as a whole. We need to target all of those hallmarks, hence the need for combination treatments, as usually single treatments only maybe affect 1 or 2, or possibly a few more of the hallmarks, but certainly not all of them together. And you can see the hallmarks here, from genomic instability all the way down to immunoaging. And as they say here, those marked with a plus have a life-prolonging therapy, and ones with a minus don't have one as of yet. Next, in the first table, they show all of the monotherapies, or the single therapies aimed at different aging hallmarks, most effective in mammals. So here you can see the first one, telomerase gene therapy affecting telomere attrition. They give a brief description of the biological effects which were studied, and also the medium lifespan, so like the average lifespan, and also the maximum lifespan. As you can see here, for 1-year-old mice, I guess given the treatment is 24% medium lifespan increase, and a 13% maximum lifespan increase for the mice given it at 1 year of age. And there are quite a number of these single interventions. And what I quite liked about this paper, taking a look at fisetin here, is that they do mention if the studies haven't been properly replicated. So in the case of fisetin, there was a study which showed it increasing lifespan by 11% as a maximum. However, this was not replicated in the interventions testing program, which is the gold standard of lifespan studies. However, they do include it here regardless. You can see here rapamycin appearing numerous times, as it's one of the most studied, if not the most studied, effective lifespan extending treatment so far. Anyway, I won't go through all these. You can see a long list. This is, as I say, this is quite a long paper, but certainly a very good reference point, something to come back and refer to in the future if you sort of have any questions or wondering about what the current or most effective treatments are. So those are all the single ones. Next, they looked at combination therapy in mammals. Once again, mice, if not entirely, then primarily. And once again, they mentioned the therapy. Actually, the reasoning behind it, which targets, which hallmark it targets. So here, this one targets 3, the biological effects, the effect of the combination. So in this one, was metformin and rapamycin, which was tested a few years ago in the interventions testing program, and at that time had the largest increase in lifespan. So saying synergistic, except female maximum. And so what the separate interventions did, so in the ITP, metformin only increased lifespan in men, the male mice, not the females, well, only by a little bit, whereas rapamycin had a much bigger effect. And combined, the maximum of the medium lifespan was 23% compared to a maximum of 21% for rapamycin on its own. And for the maximum, it didn't really make much difference for metformin, only a 2% increase for the men. Rapamycin had either 11 or an 8% increase. However, together, combination of metformin and rapamycin produced a 17 and 10% increase for females and male mice. So that was the highest scoring one so far. That was part of this gold standard. There are other interventions which have higher numbers, greater extension of lifespan, but however, they haven't necessarily been replicated. They also might involve smaller numbers of mice. So you always got to be careful, as we saw in the previous study, whether or not or how many mice are involved and whether it reaches statistical significance. So here, actually, rapamycin and acarbose, also in the ITP study, is currently the highest life extension. If we scroll to the right, it reaches up to 24% in males, which you can comparing to 17% in females for rapamycin and metformin. So currently that's the best sort of 100% validated lifespan extension. Now, there are numerous other ones mentioned here, include senolytic treatments such as dasatinib and quercetin, various transgenic mice, like this P53 TERT and P16 ARF, and also mentioning the epigenetic reprogramming here using 3 of the Yamanaka factors, OCT4, SOX2, and KLF4, and also including things such as calorific restriction, shown by the CR here. Next, in table 3, they showed other combination treatments aimed at the hallmarks. However, these did not show additive or synergistic effects on mammalian lifespan. For example, this first one here, lifespan did not exceed the effect of the separate interventions, PROP1 deficiency and GHR knockout. So again, sort of a genetic approach. Quite a few here saying no effect. Lifespan did not exceed the effect of the separate interventions. Table 4 now, they have some more combination therapies aimed at the different hallmarks, but these were done in progeroid or disease animal models, so not sort of normal mice that live their natural lifespan. So progeroid are mice which have shortened lifespans, and there's always a question, does it really encapsulate proper normal aging or not? Anyway, they also mention all the various studies here as well that affects them. And then finally, in table 5 here, they show some combined therapies aimed at the different hallmarks, but they're without any data on lifespan extension. So all these treatments had effects, but for whatever reason, the scientists, probably for money reasons, because it is expensive to conduct a lifespan study. Mice cost a lot to be kept in proper conditions, especially if there are lots of them, to make it statistically significant. And you can see the various other interventions here that hopefully in the future people will conduct lifespan studies on them. Now, to put this into a visual format, they have a nice, let's get down to the bottom, a nice little graphic here, which shows the various different hallmarks in the middle here, and then some of the treatments sort of which affect those hallmarks surrounding them. And then they propose a few different multi-combination treatments which could possibly affect most, if not all, of the hallmarks. And what's interesting here is that the green one, navitoclax, HSCT, TERT activation, and rapamycin, is currently being tested by the Longevity Escape Velocity Foundation. And I believe they've just finished their lifespan study, but they still need to analyze the data. But there are a few different suggestions by the authors of this paper as to the best way forward for combination therapies. And in fact, they propose 5 different strategies. The first is simply to combine the 5 most effective current treatments. The second is to combine the smallest number of treatments which can cover the most hallmarks. The third is to start with an existing combination such as rapamycin and acarbose and then expand upon it, sort of add extra combinations to that. Fourth is to synergistically screen the best pairs of combinations. And finally, they propose combining those treatments which have been shown to be safe in humans, such as taurine and glycine, for example. Anyway, this is a great reference paper, and be sure to bookmark it for future reference. And now for a quick look at ARDD 24, the Aging, Research and Drug Discovery meeting, which takes place in Copenhagen every year. It is by far the biggest and most important longevity conference of the year. And taking a look at their website, you can see just how big a conference it is, with a huge lineup of speakers. In fact, if we scroll down here, you can see a list of all of the speakers along with their photos. And if you follow it all, longevity science or longevity research, you will notice a lot of familiar names. It is a who's who of aging researchers, each of them giving talks or taking part in panels. It's not only researchers, but predominantly researchers. Now, Lifespan IO did a write-up of the conference, authored by Arkady Mazin. It gives a snapshot of what took place in the conference with highlights of some of the talks, photos of some of the slides, and descriptions of what went on. Now, of course, this is such a huge conference that it only can give a very small sliver of all that went down, but still is well worth your read. And of course, you can find a link to this article below this video. If you're interested in finding out more about the talks, fortunately, ARDD records all of them and puts it on their YouTube channel, which we'll take a look at. And at the time of this recording, they had just started uploading the talks, and you can find them, well, on the home screen here, but also under playlists. It says here longevity medicine track, ARDD 2024. There are a few different tracks, and I guess they are starting with this one. At the moment, there are only 6 videos available, but all of these have been uploaded in the last 24 hours, and they will probably upload the rest over the next couple of months. So do please check back. And not only can you see the talks from this year's conference, but they also have all of the talks from the previous conferences. So you can see that last year, ARDD 2023, there are 74 videos, and that gives you some indication as to how many videos we can expect to see from this year's conference. And as I said at the beginning, these are the premier aging researchers out there, so it's well worth your while to take a look at least some of these videos. Now, last but not least, is the Canadian content study. This study comes from Western University in London, Ontario, and features some quite exciting research with lots and lots of potential. It involves the discovery of a new DNA repair mechanism found in a quite extraordinary bacteria called Deinococcus radiodurans. Probably pronounced that wrong, but anyway. Now, what makes this bacteria very special is its ability to withstand extreme amounts of radiation. In fact, it can withstand amounts of radiation 10 to 15,000 times the amount which would kill a human cell. In fact, it can withstand such extreme environments that I would not be surprised if we actually ended up finding it on Mars. Now, to be able to survive in such extreme environments and to withstand such high levels of radiation, the organism needs to have some good DNA repair, because radiation damages DNA quite effectively. Now, this bacteria has several different DNA repair mechanisms, but the newest one found is quite special and has, as I said before, a lot of potential, potentially even in humans. Now, this is a quite complex paper, and I must admit it's sort of above my pay grade in terms of understanding its full extent as it goes into the structure of this mechanism. It's basically a protein. Now, fortunately, there is a brief YouTube video where the author, Robert Sabla, I probably pronounced the name wrong, apologies, Robert, if you're watching this, explains how the mechanism works. But then there is also a article from Physics.org explaining, again, in more layman's terms, the significance of this discovery. However, we will briefly take a look at the paper itself, and there are a couple of figures or graphics inside, which I'll try to explain how it works and what makes this special. Now, the name of the protein responsible for the repair mechanism is called DDRC, and let's take a look at this graphical abstract, which they fortunately include. So here is an image of the protein in question, and here's sort of the graphical abstract of it. And these blue squiggly lines represent DNA. Now, DNA has 2 strands, and the protein travels along the DNA, and if it detects either a single strand break or a double strand break, it snaps shut, a bit like a mousetrap. Now, there's another graphic found at the end of the paper which shows this a bit better. Just go to that quickly. And you can see here in the bottom right, again, these are images of the protein itself. But in the bottom right here, see the representation of a mousetrap at the bottom. And this section is like a loaded spring, I guess, not a mouse, but a broken DNA strand travels through here. When it gets noticed, it snaps shut basically and sort of trapping this broken DNA, preventing it from causing sort of any further damage or getting even more damage, and calling in a actual sort of repair mechanism. So this is bacterial DNA, and the details here are a little bit complicated, but depending on if it's a single strand break or a double strand break, it leads to different types of repair, according to the authors. And I couldn't quite see this in the paper itself, though it does mention it in the layperson's write-up, that it also acts as a beacon. It sort of calls in other machinery, other cellular machinery to come and fix the break. Now, what also makes this protein quite special, apart from sort of, in a sense, stopping the damage in its tracks, is the fact that it sort of works independently. Most proteins in our body work in conjunction with other proteins. They are sort of part of a network. This one seems to work on its own. So when they inserted the gene encoding it into another organism, actually an E. coli bacteria, they made the E. coli 40 times more resistant to UV radiation. And the authors think that this protein might be able to be put into other organisms, even mammals, even potentially humans down the road, and make them more resistant to DNA damage. So obviously you can hopefully see how this has great implications for longevity, as DNA damage is one of the myriad of causes of us aging. So if a better mechanism can be found to repair that damage or stop that damage from getting worse, then that can only be a good thing for our own longevity. And I contacted the author, Robert Zabla, again, apologies if I say the name wrong, and asked him if he had thought about the implications for longevity. And indeed he had, and they're sort of exploring this avenue further. Now, they couldn't give any sort of details as to exactly what they're doing, but he did say that we will be exploring whether exogenous, so from the outside, outside the body into the body organism, I can say, exogenous DDRC can improve DNA damage resistance in human cells. So at this stage, we don't know, but however, the possibility exists that it can be used in humans. Anyway, it's a case of watching this space and crossing your fingers and wishing them all the best of luck in their next steps. And that marks the end of another longevity review. Please don't forget to like this video and subscribe to our channel. So until next month, bye.